So cells are grown from human tissues. Now, the idea is to somehow get these cells into an animal or into a patient and get them to reform blood vessels inside the patient. And the way that this is done is that these cells are suspended in a three-dimensional gel. So this is a gel of collagen, and it looks just like this scanning electron micrograph on the side. If you look at very high magnification, it's just a web or a network of protein fibers formed from the natural protein collagen. And we talked about collagen's role in the extracellular matrix earlier. Here, this is isolated collagen, purified, and then formed into a gel. And cells are suspended within it. That's what this picture down here is to show you. This is a high magnification. A cell in here would be almost the same size as this whole picture would be a cell. And so you can imagine each cell that's suspended in this three-dimensional material is surrounded by a webbing of protein fibers. Right? And it has a thickness. Right? So unlike normal cell culture where we grow them flat on a plastic dish, these cells are suspended within a three-dimensional gel. Now there's something else in this gel here, and that's shown here. These are particles made of the material alginate. And in section a few weeks ago, you all made particles like this. Right? We made them with food coloring in them, but you learned the process. It's quite straightforward. And imagine that instead of putting food coloring in it, like we did, you put in a protein. And you selected that protein to be one that encourages blood vessels to form. It's a protein that your body naturally uses to cause blood vessels to form more readily. And that protein is called vascular endothelial growth factor, or VEGF. It's not important, the details, but it's a signal that the body uses to form new blood vessels. We've isolated it, purified it, and encapsulated it in these tiny particles, which are then suspended with the cells in the gel. So what you have here is something that you might call a neotissue, what we would like to become a tissue when we implant it back into the animal. It's not a tissue yet. It's a collection of cells, an extracellular matrix, and signaling molecules that are arranged in a three-dimensional structure. And the hope is that if you put these back into a patient or put them back into an animal, like the mouse shown here, that you would grow new blood vessels. Okay, that's the hope.